So this will be the video for the eukaryotic organelles. In class, we filled in a table as we went through notes. If you don't have that table, you can just take notes directly from the video, but be sure to draw illustrations of what each of the parts look like. We'll start with the cell membrane. Cell membranes are found in plant and animal cells. In fact, they're found in all cells, including prokaryotes. Structurally, it's made of a phospholipid bilayer. If you look at this illustration, we see two sets of these red circles attached to these yellow tails. There's a set that runs across the top here. There's a set that runs across the bottom. Bilayer means two layers. There are two layers of phospholipids with these glycerol heads pointing outward. It is a thin, flexible barrier that surrounds the cell. It is semi-permeable, which means it lets certain things through, but keeps other things out. And the way it controls that is with these embedded proteins. These proteins will allow certain things to move through the membrane while keeping certain things out of the membrane or out of the cell. It protects the cell and it regulates what enters and leaves the cells. That is its function. Next up is a plant-only structure. There are two of those that we're going to see in the notes today. The cell wall. Plant cells do have a cell membrane. It's this thin layer that you see running around the cell here, and it will still do what cell membranes do. It'll protect the cell and control what moves in and out. But what they have in addition to that is this thicker, stronger outer layer called a cell wall. It is a strong supportive layer made of cellulose, a starch, a carbohydrate. Its function is to provide support and shape, as well as protect the cell. Animals like us, we have skeletons that provide us support. Keeps us from collapsing into a blob on the ground. Plants don't have skeletons. They need something else that provides them support that allows them to stand upright. It's the cell walls that do that. Next up, chloroplasts, the second of our plant cell-only organelles. Their structure is two membranes surrounded by stacks of membranes. So if you look at it, we have these two membranes that wrap around it. And inside it, we see stacks of other smaller membranes, like stacks of coins inside a purse. This is where you find the chlorophyll. This is where photosynthesis happens. This is where energy from the sun is captured and converted into sugars, converted into food for the plant. So cell walls and chloroplasts are our two plant-only organelles. All cells have cytoplasm. It's one of the four things that prokaryotes and eukaryotes have in common. And because all cells have cytoplasm, we find it in both plants and animals. The theme of this set of notes is comparing plants and animal cells. But again, some of these things do extend beyond just plants and animals. Cell walls, for example, will also be found in fungal cells. They'll also be found in prokaryotes. Cytoplasm, found in plants and animals, and everything else. It is a jelly-like liquid that you find inside the cell between the nucleus and the cell walls. It allows for movements of things within the cells and gives the cell its shape. Cells, particularly animal cells, are a lot like water balloons. What gives a water balloon its shape is the water that's inside of it. Pop it and let the water out, it flattens out and it loses its shape. Animal cells are very much the same way. They're full of cytoplasm, and that cytoplasm gives it its shape. Pop the cell and let the cytoplasm out. The cell loses its shape, it flattens out into nothing. If you've ever had frozen strawberries, you know they're definitely not the same as fresh strawberries when you thaw them out. When you thaw frozen strawberries out, you just get this mushy, jelly-like mess. That's because the cells have been popped. When the ice crystals form inside the cell, it pops the cell. And then when you thaw those strawberries, it lets the cytoplasm out, and it just becomes a jelly-like mess. The Golgi apparatus are next. It's another structure that you find in both plant and animal cells. It appears as stacks of flattened membranes. It is the burgundy one in this picture here. They're free-floating and not attached to anything, unlike these blue ones in the back that we're going to learn about in a little bit, which are attached to the nucleus. So they're free-floating stacks of membranes. They are the packaging center of the cell. They take the substances that the cell makes, modifies them, sorts them, and packages them for distribution within the cell and to be shipped outside of the cell. They make these little packets that can either leave the cell or be transported to other parts of the cell. It's the packaging center, the packaging and distribution center of the cell. Lysosomes are these small little packets of digestive enzymes that you find within the cell. They're like little sacks of digestive enzymes. They're used to break down lipids, carbohydrates, and proteins. They're only found in animal cells. And the reason why they're only found in animal cells is because animals eat, plants don't. Plants have chloroplasts to make their food. Animals have to eat to get theirs. And the carbohydrates, the lipids, and the proteins that we eat need to be broken down into usable units for the cells. Proteins need to be broken down to amino acids. Carbohydrates need to be broken down into monosaccharides like glucose. We need to break these things down. The lysosomes contain the digestive enzymes that break down those foods that we eat, make them useful to the cell. That is the one and only animal unique structure. Mitochondria are these bean-shaped structures that we find inside cells. It's a folded membrane. That's this part with these little finger-like projections that you see. It's a folded membrane surrounded by another membrane, the smooth one you see on the outside. These are the powerhouse of the cell. They produce the energy that the cell needs. It takes the chemical energy from the food that we eat and it turns it into usable energy within our cell that will run the cell processes. Nuclear membranes are just like the cell membrane, but instead of surrounding the whole cell, it just surrounds the nucleus. And it functions the same way that the cell membrane does. It provides a protective function. It protects the DNA. And it controls what moves in and out. But this time, instead of in and out of the cell, it's in and out of the nucleus. It controls what moves in and out of the nucleus. The nucleus itself is a large structure. It's one of the largest in the cell. And it contains the DNA. It's surrounded by that nuclear membrane. Two big jobs. It controls cell functions, and it houses and protects that DNA. The way I like to think about it is a medieval city, a walled city. There's a big wall that goes around the whole city, and it controls what moves in and out of the city. That's the cell membrane. Within the city, you have different things. You have different buildings. You have places that make stuff. You have places that produce food. These are all those organelles that you find in the cell. Inside the city, you also find a castle or a keep where the king lives. The ruler lives in that keep. And surrounding the keep, you usually find another wall. It's an added layer of protection between the outside world and the ruler. This is another layer of protection between the outside world, and the DNA. The DNA is that important. You have gates along the city wall that controls what gets in and out of the city. You have gates around the castle wall to control what gets in and out of the castle. Ribosomes are in all cells, including prokaryotes. These are the protein factories. And since most cells' job is to make proteins, you find them in all the cells. They're small structures made out of RNA and protein. You will find some of them free floating in the cell. You can see some of the little specks over here on this right-hand side of this illustration. Those are ribosomes, but they are also connected to this structure called the endoplasmic reticulum. And again, they're little protein factories. They make the proteins. They are the protein-making machines. Some of them are housed on this rough endoplasmic reticulum. It is a system of membranes that are attached to the nucleus. So these membranes attached to the nucleus, speckled with these ribosomes, gives them a rough appearance. So it's called the rough endoplasmic reticulum. Think of it as the factory floor. If the ribosomes are the machines that make the proteins, the endoplasmic reticulum is the site where you find them, the floor where they're spread out and attached, where they're located. They will deliver things to the ribosomes and remove things from them for transport elsewhere. So again, you got machines on a factory floor that make stuff. So at first quality, they have machines there that make toilet paper. But the toilet paper doesn't stay at the machines. It has to be moved away from the machines to other parts of the cells. Endoplasmic reticulum does that with the proteins. You have to get raw materials to the machines. Endoplasmic reticulum does that for the ribosome. It gets materials to them. It delivers materials to the machines. It removes the end products from the machines, just like a factory floor would. You're going to have people running around moving raw materials to the machines. You're going to have people running around taking the finished product away from the machines. It's the factory floor. Smooth endoplasmic reticulum is just like the regular rough endoplasmic reticulum, but it doesn't have ribosomes on them. It doesn't have protein factories on them, so it doesn't make proteins. It makes phospholipids for the cell membranes. It makes steroids and hormones, lipids. But it also detoxifies, removes drugs and poisons, and it also acts as a storage place for calcium ions. Vesicles, those are little packets that things are transported around the cell in. They're little sacs made out of a membrane that are used to move things around both in and out of the cells. It's used to transport proteins from the rough endoplasmic reticulum to the Golgi apparatus. It's also used to move food into the cells and to take waste out of the cells. They're just little packages that things are moved around in. So again, at the factory floor, if you're making toilet paper, you just don't move individual rolls of toilet paper around. It's all packaged. It's boxed up to be moved around. The vesicles are the boxes, the packages that things are put into to be moved around the cell. Vacuoles are implants in some animal cells. They are very prominent in a plant cell. They're big bubble-like structures. You see it here and how much room it takes up inside the cell. Big bubble-like structures. It's just used for storage. It stores water and anything that's dissolved in that water, like salts, proteins, and carbohydrates. Last structure is the cytoskeleton. It's this red web-like structure that you see running through the cell. It's within the cytoplasm. It helps support and maintain the shape of the cell. It supports the organelles, keeps them in place, keeps them from moving around. And it's used for movement of these organelles when the cell divides. Again, provides some support and structure to everything. This is a cross-section of an animal cell. You see a lot of the parts we talked about. There's the nucleus. Inside it is the nucleolus. These are where ribosomes are made. You don't have to know that. It wasn't on our list. At least I wouldn't think we do. because the people that wrote the slide wrote the test. Here's our endoplasmic reticulum. You see we have some rough endoplasmic reticulum here. We have some smooth over here. This is where the proteins are made. This is where phospholipids, steroids, and hormones are made. Here's your Golgi apparatus, again, not connected to the nucleus, just out here. Proteins will go over to the Golgi apparatus to be packaged up and shipped out. They're packaged up into these red structures called, not red structures, they don't have any vesicles in here, do they? Oh, well. They're packaged up into vesicles to be transported out. There should be vesicles in this picture, but they're not. There's our bean-shaped mitochondria. There's our cell membrane wrapping around it. There's our nuclear membrane right there. Here's some free-floating ribosomes, not on endoplasmic reticulum. These red spheres, those are lysosomes with our enzymes in it. This is showing you some vacuoles being used to store stuff. Centrosome is not on our list, so I don't think you need to know it. There's a plant cell. Note the difference. It's square-shaped because it's got this cell wall around it, providing it with structure and shape. Think of it as a box that the cell sits in. It still has this cell membrane underneath it. So even though it has a cell wall, it still has a cell membrane. There's your big dominant vacuole. Biggest thing in an animal cell is typically the nucleus. Biggest thing in a plant cell is typically the vacuole. There's our nucleus or nucleolus. Got chloroplast this time. 2 membranes with stacks of membranes inside of it. Still got mitochondria because that sugar that it makes has to be burnt somewhere. We got ribosomes in here. These have to be vesicles in this one because, again, there's no lysosomes in a plant cell. Call them amyloplasts. Again, it's not on there, don't know it. There's our endoplasmic reticulums, Golgi apparatus, etc. I've mentioned it before in this video, you need to understand how proteins are made. The instructions for making a protein are here in the nucleus. Those get sent out to the ribosomes where the proteins are assembled. Some of those ribosomes are attached to the endoplasmic reticulum. Some of them are out there free floating in the cytoplasm. The proteins are carried to the Golgi apparatus once they're made in the vesicles. Remember, you need to make RNA to send the code out of the nucleus because the DNA never leaves. It stays in there. An RNA strand is made, it's sent out of the nucleus to the ribosome. It's RNA that carries the instructions out to the ribosomes. Again, once the proteins are made, they're packaged up by the endoplasmic reticulum into a vesicle, sent out to the Golgi apparatus, where they're further processed, sorted, and packaged to be sent out of the cell or to other parts of the cell. And again, they're all carried around these things in these structures called vesicles. So there you have it. That's what you need to know about the parts of a cell. Again, if it's in a worksheet we do or it's an illustration we do and it's not mentioned specifically in the table that we filled out, then odds are you don't have to know it.